Good day, everyone. I'm here to represent the life science project that I've designed. The knowledge strand of this project is life at molecular, cellular, and tissue level. The topic and the content is the meiosis. So by the end of this project, learners will be able to state the phases of meiosis and be able to describe the event that occur in each of these phases throughout meiosis 1 and meiosis 2, and be able to describe how crossing over of homologous chromosomes will increase genetic variation. So meiosis is a division of cells in sexually reproducing organisms that break down the number of chromosomes in gamete. Diploid human cells, also referred to as body cells or somatic cells, contain two sets of chromosomes, one from each of the parent. So meiosis is a process that involves one round of making DNA and two more rounds of separating the two sets of chromosomes. In the process of meiosis, there are two distinct stages, that is meiosis 1 and meiosis 2. Each of these components is composed of four phases, that is prophase, metaphase, anaphase, and telophase, which is similar to the process of mitosis. So the first phase of meiosis is by far the most complex of the meiotic division. So prophase 1 alone, it takes up more than half of the time due to its 5 substages. So meiosis 1 differs from mitosis in three fundamental ways. That is, meiosis 1 is characterized by a reciprocal recombination, the homologous chromosome pairing, and also during the process of meiosis 1, two steps are initiated in order to facilitate the release of cohesion sister chromatids. As I've mentioned that there are four phases in meiosis. Now we are going to explain what is happening in those phases. So in meiosis 1, prophase 1, the homologous chromosomes pairs assembles to form a tetrad. And this is the phase in which genetic recombination takes place. Metaphase 1 occurs which the corresponding homologous chromosome positions are aligned opposite each other. So chromosomes are randomly distributed to daughter cells, resulting in the distribution of homologous chromosomes. In anaphase 1, the chromosomes of the cell migrate to a different location, while the sister chromatids are left intact. Each cell is now composed of 23 chromosomes. Telophase 1 cytogenesis occurs when the homologous chromosome migrate to the pores, resulting in the formation of two daughter cells. So in meiosis 2, prophase 2 chromosomes began to migrate to metaphase, and there's no replication. Metaphase 2, when the two sets of chromosomes come in contact at metaphase 2 plate. In anaphase 2, sister chromatids are separated from one another and migrate in opposition direction. Lastly, in telophase 2, cytogenesis happens and four new cells are created, each of which possesses 23 chromosomes. So with the project that I've designed, I used a microscope to observe various stages of meiosis using different types of cells. The result has shown that there are indeed various stages of cell division when using different types of cells observing under the microscope. And this is because of the position of chromosomes. So the meiosis topic is very relevant to the FET phase learners because in order to create new offsprings, humans require the process of meiosis. So meiosis is essential for human life, for the human life. And this will help learners to acquire a scientific understanding of human reproduction through the acquisition of knowledge from a range of sources. Because it can be challenging for learners to comprehend how homologous chromosomes assembles and how crossing over occurs and how gene replacement occurs, the teacher will implement the hands-on activity, and that is, will want learners to construct a model of homologous chromosomes. So learners will construct the model of homologous chromosomes to be able to visualize the concept and the steps that are happening in the process of meiosis. So learners will collect the materials that will be needed to construct the model. That is the 2 pieces of paper. One will be in pink and one will be in blue. These different colors is just to differentiate the chromosomes, which one will be for paternal and one will be for maternal. And they will also need a scissor and tape. So this construction of models will reinforce the teaching by helping learners to visualize how the crossing over occurs and how the homologous chromosomes assembles. So the teacher will assess learners' knowledge by doing the question and answer method. Question and answer method helps the teacher to be able to interact with learners. And when learners are interacting with the teacher, they'll be able to see where they need to improve. And this will also help the teacher to see the level of learners' understanding of the concept. So the model that they've constructed, the marker will use the possible point to check how their model have been constructed. That means the marker will use the knowledge to see the project. The marker will use the knowledge to see if the project shows understandable of meiosis, and the presentation if it's comprehensive from the beginning to the end. And the marker will also observe the creativity of the model and also the neatness.